Sounds good.
Okay. Hi, everyone. Thank you so much for joining us. My name is Lily Cohen. I'm a first-year medical student at Mount Sinai. And along with Kala Kilnani, I run the One Health and Emerging Pathogens interest group. And I'm absolutely thrilled to welcome Dr. Anna Fagre as today's speaker. Anna is a veterinary postdoctoral fellow at the Center for Vector-Borne Diseases at Colorado State University, and she's both a fantastic scientist and a personal friend who I've had the honor of collaborating with on several projects this year. So we'll definitely have a few minutes for questions at the end, but if any arise during Anna's presentation, we would just ask that you please put them in the chat box, which Kala and I will be monitoring. With that, take it away, Dr. Fagre.
Hello, my name is Anna Fagre. I use she, her pronouns, and as Lily mentioned, I'm a veterinary postdoctoral fellow at Colorado State University, and I am also, the past year, have been working with the Verena Consortium, and that is some of the work that I'm going to present on today, and that's how I know Lily. Thank you so much for the nice introduction, and thank you to the group for having me today. I'm really excited to chat with you all about Spillback. Specifically human to wildlife pathogen transmission. So quickly, I'd like to just talk about my background to give you some kind of context. I don't know how often you all have interacted with veterinarians. I really did not get to start working closely with MDs in a professional sense until the last few years. I grew up with some MDs in my family, and so I always kind of had these ideas in my mind about how things translated between human medicine and animal medicine. But the past few years, particularly during the pandemic, I have been able to collaborate more closely with MDs. So after undergrad in Iowa, which is where I'm from, I moved out to Colorado and I joined the MPH-DVM dual degree program, which is a 5-year program. And for my MPH, I studied on antimicrobial resistance, biosafety, and biosecurity. And I really was kind of most interested in global health and wildlife disease ecology and pathogen sharing between wildlife, livestock, and humans. And that interest began during undergrad. I was doing some work over in Tanzania and got really interested in how livestock health really directly impacts the health of the Maasai communities that we were working with over there. And so I decided if I was able to do something like an MPH, it might give me some more exposure to global health. So I did this antimicrobial resistance focused MPH. And then when I moved into vet school, I got a bit more focused on global health, zoonotic diseases, and wildlife conservation, and trying to figure out really how to marry or kind of straddle these two fields of wildlife conservation and public health, which can seemingly be at odds with one another when we discuss things like emerging zoonotic diseases that originate in wildlife. So after the DVM MPH program, I matriculated into a 2-year residency at the Veterinary Diagnostic Lab here, and that was heavily focused on infectious disease diagnostics. So more the kind of clinical microbiology, and then being able to work with DVMs. And I also worked with some MDs during this time, had some conversations with them, especially when we had public health related cases. We worked closely with the Department of Public Health, working up, rabies exposures, and we had some plague and tularemia. And there are a lot of kind of unusual and interesting zoonotic diseases out here in Colorado. And so we were able to work with the Department of Public Health during those cases. And that was really interesting to me. And I knew I wanted to do something kind of at that interface of human and animal medicine, particularly related to infectious disease, maybe diagnostics. I was really interested in surveillance, and I was able to more closely investigate some of these questions surrounding wildlife disease ecology using surveillance approaches and also hypothesis-based approaches during my PhD. So my PhD, which I just defended, I guess, a month ago tomorrow, March 22nd, I defended. So I haven't been a PhD for very long, but my PhD was a long stretch, and it focused on bats as reservoir hosts for emerging arboviruses. And the shift from arboviruses and kind of, broadened out to coronaviruses during the pandemic, as it did for many individuals. And it's been a really rewarding and fruitful experience. I've got a couple pictures here. So this is during vet school, working over in Nairobi with the veterinary students over there. Here I am a couple years ago in Tanzania. This is a mosquito trap. And so we were looking at mosquito ecology and trying to kind of get an idea of which hosts mosquitoes were feeding on as a way to analyze the risk of Rift Valley fever virus circulation. And Rift Valley fever virus is a Bonia virus that has not been introduced to the United States, but it's considered a concern for potential introduction, and it impacts livestock and humans and wildlife, and it's transmitted by mosquitoes. And so this is what we were doing here. And then this is me reading a plaque assay. I believe this is me counting orbivirus plaques for a new orbivirus that we discovered and characterized. And so there's been some kind of viral discovery stuff that I've been involved in as well, which has been a lot of fun. And I would like to mention here, during my PhD, I did a 1-year translational medicine fellowship. And so that was really cool. It was housed down at we have a medical school in Denver. And so I went down to Denver a couple times a month, and I worked with the neuro ID residents and fellows there, and got to kind of learn about how they worked up cases. And a lot of them were, arbovirology or arbovirus rule outs. And so that was really interesting to get an idea of, okay, these viruses that I have been studying the ecology of through, looking at the vector distribution, and also through doing some in vivo work with our captive bat colony. Now I can really see how they might manifest in human patients. And so the kind of translational comparative aspect of that was really interesting to me. And I think programs like that are, I hope that they increase in frequency after the pandemic. And I'll talk a little bit about those in a few minutes here as well. And then after, let's see, I guess in a couple months now, I'm joining EIS. And so that starts in July. And EIS is the CDC's 2-year kind of boots on the ground disease detective fellowship. And so it stands for Epidemic Intelligence Service. And I don't know where I'm going to be stationed yet, but right now we're going through a big match program, and there are a lot of MDs in that program. And I don't know if y'all have heard about that program, but if you're interested in public health, global health, epi, I highly recommend looking into it. There are just ample opportunities and really diverse opportunities to get involved in some of these questions, but there are also, occupational health, chronic disease. There are also positions in those branches as well. So there are a lot of cool things you can do in EIS. So I highly recommend checking that out. Feel free to reach out to me if you have questions about that. So getting into kind of our story today, I'm sure if you're in this group or in this talk, you probably have some interest in this idea of pathogen emergence and increasing pathogen emergence in recent decades largely due to climate change and human activities. So land use change, driving habitat degradation, and ultimately resulting in biodiversity loss, and also driving an increasing interface between humans and wildlife, whether that's, at this forest edge where there's more interactions, or people harvesting animals for protein, or just interacting with them for any number of different reasons. We are seeing more interaction between humans and wildlife due to their inability to kind of exist peacefully away from us. And so when we think about this in terms of what does this mean for public health, we do know that over 75% of emerging viruses are zoonotic. And this is a typo, I apologize. And the majority of these originate in wildlife. And so when I say originate in wildlife, it doesn't always look like, okay, a person eats an animal or a person interacts with an animal and then this turns into an outbreak. Sometimes there can be intermediate hosts that are livestock. So the virus moves from, so for MERS coronavirus as an example, the virus moved from, probably had its evolutionary origins in bats, moved into camels, which tend to interface a lot more closely with humans than bats do, particularly in the area of the world where we saw that epidemic take off. They're used for agriculture and all sorts of other purposes. And so we sometimes see this intermediate host, but we consider the virus to originate in wildlife if it has its evolutionary origins in wildlife, like it did with bats, former coronavirus. And that's just one example. I mean, we see outbreaks of filoviruses resulting from spillover events. I don't have the word spillover on this slide, but that is kind of colloquially what we use to describe these events. And so filoviruses, coronaviruses, henipaviruses, Hendra and Nipah have been in the news a bit lately, many strains of influenza. And so there are a number of different pathogens that we appreciate the impact of this. And so this is a nice figure by Christine Kreuter Johnson and some of the folks at UC Davis and their collaborators. And I really like this image. I think they did a nice job of providing this idea of, okay, how does a virus, particularly viruses with high host plasticity or the ability to adapt to novel hosts rapidly, like we appreciate with coronaviruses, how they move over into either an arthropod vector or a vertebrate vector, vertebrate host. We often call these reservoir hosts. And when we talk about reservoirs, we usually think of the animal, the wildlife reservoir, not having its health impacted, or at least suffering minimal morbidity, mortality, and not having that pathogen really impact its ability to thrive and reproduce. And this isn't, a hard and fast rule, but generally when we discuss reservoir hosts, that's kind of what we think of. So this figure here talks about the virus moving from the vector, and then this spillover event where it moves into a human host and then is able to, due to its high plasticity and ability to mutate rapidly. Generally, these are RNA viruses because they tend to evolve more quickly due to proofreading enzyme issues or enzyme proofreading issues. And so then we see this amplification, this human-to-human transmission. And sometimes in the case of COVID-19, we see global spread. And so in the case of COVID-19, there has been some increasing conversations about, okay, now that the pathogen has reached a high enough prevalence in human populations, will we see it spill back into animal populations, whether those are captive populations or wildlife populations? And so this idea of spillback, which mirrors spillover in a way, has been at kind of the forefront of everyone's minds, and we have seen it impact animal populations. And so that's what we'll be talking about today. So thinking about some of the specific concerns surrounding spillback of SARS-CoV-2, spillback is kind of the colloquial term we use. Zooanthropogenic transmission is the big long fancy word for it. So you might see zooanthropogenic transmission referring to spillback here. And so we did have confirmed zooanthropogenic transmission early on in the pandemic. And we saw this in domestic animals in Wuhan. We saw infected cats and dogs. We started to see infected cats and dogs in other countries where the prevalence of human disease was high enough that it reached that threshold and it was able to spill back into populations of animals, particularly those that interacted with humans at a very close and frequent interface. And then another example of that would be in zoo animals. We saw some instances of spillback. And so the interaction with zoo animals is going to be different in terms of proximity and frequency of interaction, but with a virus that is fairly transmissible and is able to aerosolize, you don't need to have the animal, sleeping on your chest at night for that transmission event to occur. So we appreciated that with non-human primates, big cats, lions, tigers, and then more recently, I think at the Georgia Aquarium a few days ago, they reported it in some of their otters. Now, thinking to other examples of this, and maybe the most notable example, or the example that's gotten the most attention, has been in mink on fur farms, particularly in Europe, but also here in the United States. In Utah, we saw huge outbreaks, and we saw the pathogen spill over from humans into mink, and then actually we saw a spill back from humans into mink, and then we actually saw a secondary spillover of the virus from mink back into humans. And so we'll talk about that in a second here, but I'd like to kind of talk about why we were so interested in looking at the potential for spillback in wildlife species. And there are a lot of unknowns when we look at the risk of pathogen transmission either from or to wildlife species. And so it's, often prudent and best practice to act out of an abundance of caution, especially when some of these species are, endangered or threatened, and are facing other threats like habitat loss from land use degradation. So we saw early on in the pandemic, we saw a widespread moratoria on bat research and bat handling. And that was the result of the IUCN and other groups kind of having these, extensive dialogues and really thoughtful conversations about what the risk might be. And obviously your risk analysis changes as you learn more about a pathogen. And so this was a very iterative process. It's still ongoing in terms of risk analysis and trying to figure out maybe which activities we should monitor more closely, which activities we should change, and which we should halt altogether. And so there was also concern about habituated non-human primates. And so habituated non-human primates are going to be those animals that are in close proximity with humans. So thinking about some of the mountain gorillas in Rwanda is a good example. And when their populations are particularly threatened, they will be protected by park rangers and by groups in kind of preserved areas, and they're closely monitored by veterinary staff, they're closely monitored by the rangers, and this is a way to protect them from poaching, and but also, due to this kind of increased interaction and increased proximity, there have been documented instances of disease transmission from humans back into non-human primates. We've seen measles outbreaks. We've seen multiple respiratory viruses move from humans into non-human primates, both in these habituated circumstances, which is kind of almost free-ranging, and also in captive primates. We've seen this pretty extensively. So in comes Verena. Verena is the Viral Emergence Research Initiative, and this is a group that I joined last spring, and it's headed up by, I guess I should have names here, Colin Carlson, and Colin is at Georgetown, and Greg Albury, who's our program coordinator, is also a postdoc at Georgetown. So Colin's a professor and Greg is a postdoc, and they were having these conversations about spillback and, what is the real risk? And my ears perked up, and I said, hey, I'm interested in that. And some other folks' ears perked up, including Lily's. And this is when I met Lily. I'm sorry, Lily, I should have checked if that picture, I see you covering your face. I think it's a cute picture of you and the goat. So this is when I became closer with Lily, and Lily's now become a dear friend and colleague. And we also had help with many other people. We pre-printed this last week, so you can find this online, and I'm happy to share the link, or you can Google it. It's on EcoEvo archive preprints. Excuse me. And so this is a massive effort, lots of folks involved, lots of thoughtful conversations around how we interpret some of the data that exists, and then try to highlight some of the existing knowledge gaps. So what we really were interested in is we looked at what we know about SARS-CoV-2, since that has been, the most recent sort of, really intensive conversation around spillback. Sometimes you'll see things in the news like, chimpanzees caught measles from humans or something. But I mean, this pandemic really brought the idea of spillback and the idea of pathogen transmission back to animals, really brought it to the forefront. So thinking about kind of the different steps that are undergone for spillback to happen and for a secondary spillover event to occur, we used SARS-CoV-2 as a model. And I just want to touch on this before we move into kind of the more generalized evidence base of spillback throughout history, because I think it's interesting to not only to look here at some of the things that have happened with SARS-CoV-2 over the past year, but to think about this in terms of a bit of a risk analysis. So, the risk of spillback, thinking about this through the lens of what is contact frequency. Companion animals, high. And then we have things like farmed mink, which, mink are generally not domesticated animals in the sense that we, use them for livestock or companion animals, but we interact very closely with them when they are farmed. And so thinking about stressors that animals face when they are housed in captivity, when they are housed in very concentrated, stressful environments, and how that suppresses their immune system. I think it's really important to kind of contextualize all of this. And then we also kind of thought about, so enzootic potential. Once that spillback event does happen, how likely or how easy would it be for onward transmission to animals of the same species? So thinking about a farmed animal, it's going to be very high because they're very intensively housed, generally speaking. And then a mitigation effort required to prevent the secondary spillover back into human populations. We considered this to be high for farmed mink because humans are working very closely with these animals, and depending on biosecurity practices and whether folks at these farms are wearing masks and gloves every day, there are just a lot of considerations. And so we kind of did this thought exercise for intensively managed farmed animals like farmed mink, and then captive wildlife in zoos. And we also highlight documented evidence here. Synanthropic wildlife, or those wildlife that are very close to humans. So thinking about the escaped mink, they found some mink that had escaped from these fur farms that were, they detected some nucleic acid from SARS-CoV-2. And then lastly, thinking about the potential for a novel wildlife reservoir to establish following a spillback event. And, the risk is going to be quite low depending on the contact frequency, but assuming this is a truly free-ranging animal species, the risk will be low, but then the mitigation effort required after that pathogen sets up shop and is able to undergo enzootic or kind of ongoing transmission and circulation, this mitigation effort would be quite high. So that's kind of the lens through which we were thinking of all of this. So I'll walk you through a figure, and this figure was helpful for us to kind of conceptualize what we were looking at in terms of the different kind of risks of this event happening. So from the original reservoir, which, we hypothetically used a mouse here, they are reservoirs of hantavirus. So I guess if we wanted to think about hantavirus, we could. We didn't put a bat here because everyone thinks of bats, and quite frankly, they get enough flack. So we put a rodent here. And so moving from the original reservoir into establishment in humans. So there's your spillover event. And then it will undergo, or could undergo, spillback. And then this is when, after establishing in humans, it moves into a new animal host, a new species of animal. And so we kind of thought about this through the lens of two different potential pathways, which each pathway being presenting one of the major risks. So if we think about the major risks of spillback, one of the big concerns that's been discussed is, could this disease being introduced into a free-ranging animal population have conservation impacts? So could it result in substantial host morbidity and mortality in this new population? And then the second potential thought, or kind of the second concern, are concerns related to public health. So if there is a new maintenance reservoir set up and it begins to undergo enzootic circulation in this new maintenance reservoir, then could we see a secondary spillover event back into human populations? So effectively, the new maintenance reservoir would continue to source additional human infections. And if something like that would happen, it would just be, it makes control of the pathogen really difficult. And so this is kind of the schematic that we've used, thinking of pathway 1, the conservation impacts, and pathway 2, the public health impacts from the establishment of a secondary reservoir. So we looked to the literature to see if other folks had kind of explored this question. And in 2014, a group out of Florida had put together systematic review of some of these zooanthroponic or spillback events. And there weren't that many wildlife associated events in this review, but we wanted to use this as kind of our starting point, since this is really the only review that existed on spillback to date. So we began to extract data from the literature. And so thinking about pieces of information that we took from each paper, we pulled, which wildlife species are impacted. Sometimes it was more than one. We wanted to look at the pathogens that were present in those populations, or those pathogens that moved from humans into animal populations. And we focused on molecular detection, so PCR generally, or actual isolation. So isolation of live virus or bacterial culture. And I suppose antigen testing would also fall in this category of what we were interested in, but the bulk of results were either PCR or, isolation of virus. And the reason I'm touching on this is because we opted not to include serology, because with serology, it can be very difficult to kind of delineate and characterize transmissionality. And so we discussed this a bit more in the paper, how we decided to rule these out. If you're interested in serology, I can provide you with the papers that had serology as their diagnostic parameter, and we can talk about that. But we opted not to include them in our evidence base for the sake of consistency, and also kind of for the ease of delineating transmission directionality. So we were also interested in looking at, so the animals affected, were they free ranging, were they in zoos or maybe captive rehab facilities, or were they captive, like owned animals, like Joe Schmo goes and has a pet monkey that was actually free ranging and now is a pet, which read a couple of those, and very upsetting to read. And so there were a couple of these as well. And then whether or not, almost most importantly, one of the most important things I think is whether or not those animals experienced morbidity or mortality. So after spillback, were the animal species adversely impacted, or was this just routine surveillance and they were, a research group was doing PCR on a bunch of captive skunks and just happened to find influenza, which, was the case for some. And then we were also interested in trying to analyze whether or not onward transmission occurred. So if animal A was infected, did they also find it in animals B, C, and D later, or was the N just one? Was it just a single animal? So we were interested in both intraspecies transmission, so animal to animal, or the presence of, okay, this particular group of animals was infected by a pathogen spilling back, and then they were able to source additional secondary spillover back into human populations. So those were some of the things that we looked at for each of the papers. And then getting into the evidence base, there were 104 animal species represented. We ended up with 97 papers. And some of these papers described multiple pathogen groups or multiple animal species. So that's why these numbers don't necessarily add up. We don't have, 97 papers describing 97 animal species, because some of these were reports of tuberculosis spreading through a zoo, for instance. And so there were multiple animal species impacted in those reports. But as you can see, there's quite an abundance of different pathogens. So 8 bacterial genera, that should say, 10 viral families. We had fungi, we had parasites, and it was quite diverse. But I think what's quite interesting is that of these 97 studies, over half of them described spillback in primate populations. And, there are multiple reasons that could be. It could be these animals are monitored more closely because they're large, particularly, large and charismatic, particularly those that are habituated and have eyes on them all the time, or in zoos or captive facilities. And it could also be that there's often more pathogen sharing between those species that are more phylogenetically related due to similarities in our receptors and our cells that allow for similar tropism of some of these pathogens. And so another interesting thing is that when you looked at the non-primate records, over a quarter of those non-primate records involved mycobacteria and tuberculosis. And again, these numbers aren't going to add up because some of these studies had primates and non-primates. And I do have the tables that I have as slides at the end if you're interested with all of the breakdown. Otherwise, you can just pull the paper and we can talk about that if you're interested. But yeah, I thought it was pretty remarkable that we saw such a burden of tuberculosis in the non-primate reports. And if you think about it, I mean, if tuberculosis potentially originated in humans, and so like, is tuberculosis the original spillback? We've seen tuberculosis in elephants. We've seen tuberculosis in not only primates, but also several other species. We see this spillback of mycobacterial pathogens into animal populations. And then one thing we talked about a lot is there was a remarkable lack of representation of bat and rodent species in our evidence base. And this is interesting if you think about the fact that 1 out of 5 species are bats. So there are over 1,400 species of bats, over 2,000 species of rodents. And so when you look at the proportion of overall mammalian or overall vertebrate diversity that bats and rodents provide, it's quite interesting, I think, that we haven't seen more reports of spillback in these species. And so thinking to why could this be, I think it maybe is just that we're not surveilling them for pathogens that maybe have spilled back. But that also to me is a head scratcher because there's been so much pathogen discovery effort. There have been so many pathogen discovery efforts in bats and rodents. And so this is really interesting. One thing that I don't know if we talk about this in the paper. I think we do. I think there's a line about it. But often when we see these reports of, okay, we found pathogen X, Y, and Z in these wildlife populations, and they have zoonotic potential, it's like, oh no, this is bad. These animals that live, on the outskirts of town have lepto, and that's bad because humans can get lepto. But I feel like often the conversation is lacking this question of, okay, how did that pathogen get into the wildlife population in the first place? So again, some kind of like not philosophical, but yeah, it's kind of some head scratchers still. I think that this study has prompted additional questions. And so looking at our schematic here, we focus on kind of interpreting these results, interpreting these numbers by looking at pathway 1, so the conservation impacts, morbidity, mortality, and new hosts, and then pathway 2, those numerous, or if there are numerous, secondary spillover events that might occur from the establishment of a new maintenance reservoir. And so for this would be characterized by ongoing, oh, I lost my laser pointer. That's sad. I'm sure it'll wake up. Okay. For this new maintenance reservoir, the idea would be, or we would characterize it as ongoing transmission of this pathogen in the new animal population. So this was kind of the second scenario that we interpreted. So thinking about what the threat of spillback is to conservation, so looking at reports of morbidity and mortality following spillback, over half of all documented spillback events did result in morbidity or mortality in the host. But of these 61 that described morbidity and mortality, 50 of them were in captive and habituated populations, so zoos, owned animals, rehab centers, things like that. And very few reports, only 11 of those 61, described truly free-ranging animal populations. And that to me is just, this was, I think, surprising to a lot of us. And it could, be explained by the fact that perhaps if there are morbidity and mortality events in free-ranging animal populations, those animals get scavenged and they just get missed. So it's hard to know if, lack of absence, or I'm sorry, if absence of this phenomenon indicates a lack of its presence, or is it just we're missing it, we're not reporting it, we're not detecting it due to scavenging and other things. So this was the fun part, was kind of looking at the numbers and trying to identify patterns or potentially interpret them in light of what this means for what are we missing. And so when we look at the International Union for the Conservation of Nature, and we look at the red list of threatened species, we wanted to see for those animals that were free ranging that were impacted, what was their kind of conservation status, I guess. You know, are they threatened? And if they are threatened, are they threatened by infectious diseases? So the red list of threatened species provides for the animals that have been evaluated and have enough data to be evaluated, they look at the status of those individuals in the wild, and they also rank or kind of score different threats to those species based on peer-reviewed literature and data from the field. So, you know, how big of an issue is habitat loss? How big of an issue is poaching? How big of an issue is infectious disease, for instance? And so we wanted to look at this question, and nearly a quarter of the free-ranging animals impacted by spillback were endangered or critically endangered. And so I didn't include vulnerable here, focused on these two. And so 13 of those species fell into critically endangered or endangered. And of those 13, 6 of them were considered threatened by infectious disease. And most of those, all but one of those, were primate populations. And so the idea there being spillback. So primates in sub-Saharan Africa are impacted by Ebola, for instance. And then the non-primate species was African wild dogs, and we see them impacted by rabies and distemper moving from captive dogs into their populations and things like that. So this was a way for us to kind of contextualize what the risk of these spillback events might be to wildlife conservation. And for most of these species, habitat loss was listed as the most significant threat. But I think it's important to consider that it's really difficult to analyze some of the kind of synergistic drivers of biodiversity loss. So thinking about habitat loss in conjunction with infectious disease, and, you know, could infectious disease be the straw that breaks the camel's back when these populations are already severely fragmented and compromised. Now, thinking to our secondary spillover occurring from our new maintenance reservoir, no studies actually supported maintenance of a pathogen in a new free-ranging wildlife species, which we thought was quite interesting, and really, that was kind of one of the like whoa moments for us. And there's only one instance of secondary spillover that was confirmed. And I should have mentioned earlier on, these 97 studies, we did not include any SARS-CoV-2 studies in those 97. So we wanted to look at the kind of before SARS-CoV-2 and non-SARS-CoV-2 spillback. What else do we know? So this particular paper, or this particular report, is not included in our evidence base because the only instance of secondary spillover from maintenance of a pathogen has been American minks in fur farms. And so this has been documented in a couple of places. So in Utah, they found escaped mink that were positive, and a preprint just came out of Spain 2 weeks ago, right before we pre-printed this, so we had to add a line about it. They actually found SARS-CoV-2 nucleic acid in mink that were quite far from fur farms in Spain. And so it really raises the question, does this pathogen have the ability to set up shop and undergo, you know, enzootic ongoing circulation in some of these mustelid populations, mustelid being minks, weasels, ferrets, that seem to be really susceptible to this pathogen, and they're very cute. And then we also discussed some of the, you know, how can we try to better investigate and understand spillback? And so we went through some of these thought exercises, and I think these are good opportunities. There are some opportunities here for collaboration between DVMs and MDs. So during SARS-CoV-2, I collaborated with many MDs and scientists in industry, and I got to help out with some preclinical drug trials, so investigating the impacts of investigating the impacts of antivirals on the reduction of SARS-CoV-2 pathogenesis using like a hamster model. We also investigated the potential for wildlife to serve as reservoirs or as hosts for SARS-CoV-2. So we preprinted and are trying to get published, it's under revision, this paper where we established ongoing transmission of SARS-CoV-2 in deer mice. And so studies like this are really great opportunities for interdisciplinary collaboration, which I'll talk about more in a second. But I really like this figure that Greg put together because I think it does a good job of talking about each of our different kind of 4 approaches, and then some of the advantages and drawbacks of each of these approaches. So for laboratory experiments, for example, we can manipulate them, we can easily control them, but are they really realistic in terms of what the animal would be undergoing stress-wise in the wild, etc. Incidental captive infections, so species X gets infected at a zoo, your pet dog gets infected with SARS-CoV-2, something like that. So kind of the canary in the coal mine idea. These are more realistic exposure situations, but we don't see this happen very often. And really, aside from captive animals where maybe you're kissing your cat or dog or something, this is going to be largely dependent on either environmentally persistent pathogens, so Giardia being transmitted in water, or an aerosolizable pathogen. So some of the questions that we can answer with lab and incidental captive infections are just getting an idea of susceptibility. So can the pathogen infect the host? And then looking at whether or not the host can shed the pathogen. Is there onward transmission of the pathogen either to another animal of that same species, to another species, or back to humans? And then what kinds of impacts do we see in the animal? Does the pathogen make the animal sick, or was this household surveillance looking for SARS-CoV-2 in the kitties of people who were infected, and we just happened to find SARS-CoV-2 nucleic acid in these cats and they seem fine, asymptomatic? So, and then also what we talked a bit more about today are these incidental wild infections. So, more realistic conditions here, but difficult to observe for a number of reasons that we talked about. And we can answer some of these questions, but I just think there are a lot more limitations and barriers that we face in our ability to answer these questions. And then getting to kind of the bread and butter, or some of the bread and butter of what Verena does, less of what I do, is revealing patterns, which, we all did that, but then trying to use these data to generate some predictive models and get an idea of whether or not spillback will be likely in specific animal species or in specific geographic regions. And so there are multiple tools and data sets that are used to answer questions like these. And this is kind of where we're hoping we'll see this go, because there's been a lot of predictive modeling surrounding spillover and, which wildlife species should we be looking at? Which geographic hotspots are there that we should be concerned about for spillover, but we don't see the same for these spillback processes. So then, 10,000 foot view. So what is the threat of spillback to conservation and public health? For most populations, I don't want to generalize because infectious diseases do play a large role in the decimation of non-human primate populations, like Ebola virus, and we've seen measles virus impact non-human primate populations. But for the most part, habitat loss and other anthropogenic stressors, climate change, are more significant threats to a lot of these species than infectious disease. And I think that we need to really, as a scientific community, reevaluate how we're looking for these events. So when we saw how many of these, and you can look in the paper, we really outline it kind of by species, and we saw lots of spillback described in primates and elephants and, some of these really large charismatic species. And so are we missing spillback that is happening in bat populations, rodent populations, peri-domestic kind of synanthropic, like raccoons and skunks and things like that? So I think we need to really kind of reevaluate if we do want to get an idea of how often this is happening and which pathogens this is happening in. I think we maybe need to shift the way we're doing some of the surveillance. Difficult to characterize, transmission directionality is complicated unless you're using, unless you're using molecular epidemiology or, I did not mean to do that. Molecular epidemiology, and you're tracing the spread of these pathogens using, molecular techniques or other ways to really try to delineate the movement, like network models, things like that. It's really difficult to establish directionality. And then one of the big questions is, are spillover and spillback symmetrical processes? And there's a great figure that Greg also made in the paper, and it shows, thinking about the transmission modes, there are definitely considerations that suggest maybe they're not exactly symmetrical processes. So thinking about ingestion of, humans eating wildlife don't frequently eat humans. So that's not going to be as symmetrical when we think about modeling the risk or analyzing the risk of these processes, but something like an aerosol exposure is going to be less unidirectional, right? Like everyone's exposed. So just thinking about the transmission mode is really important as well. And then, using some of these ideas, what do we need to do, and what kinds of data do we need to generate to use predictive modeling for spillback like we do for spillover? And really quickly, I just want to talk about how, I don't know how much y'all have heard about this idea of One Health. We talk about it a lot in vet school. And, we see it as a way to increase collaboration between MDs and DVMs. And I think there's a lot of really exciting opportunities there. And I think we've seen strengthened collaborations during the pandemic. So touching on like what I recently said, some of this translational kind of comparative biomedical research using animal models. I worked with MDs for this. Vaccine distribution, we're now seeing veterinarians vaccinate people alongside MDs in many states. And then some of this, the epidemiologic tracing of cross-species transmission. So we're seeing a lot of groups do like household transmission studies, testing animals, testing humans. There's been a lot of surveillance in zoos to make sure that zoo animals are not becoming infected. So we're seeing more opportunities, I think, or maybe a stronger interface between MDs and DVMs during the pandemic, but we still have a really long way to go, I think. And so I've just been brainstorming the last couple of years, different ideas for maybe trying to increase these collaborations and facilitate these interactions so that, because there are so many similarities in what we study. And, humans are animals too. And we study it in different species, and, humans don't have like a rumen like we study in cows, and there's going to be different nuances and things like that. But I mean, for the most part, there's such a rich opportunity for like conversations around some of these like comparative aspects of even anatomy and physiology. But when you get into disease transmission, I think it gets really interesting. We also use comparative medical techniques to look at, chemo drugs and things like that. We have an animal cancer research center here at CSU that works very closely with medical schools, looking at some of the chemotherapeutic agents that are used and limb sparing techniques for, osteosarcomas and things like that. But I think thinking about ways we can really enhance this for students during training. So maybe these are just ideas I have, but if you have other ideas, I would love to hear them. And I'll give you my email here on the next slide. But summer research exchanges, either where DVMs and MDs are working alongside each other in the lab, or maybe at public health agencies. These could also look like fourth-year rotations that co-host MD and DVM students. And then translational postdoctoral fellowships, so similar to the one that I did, like year-long, fellowship where you're still doing your own research, but you are also encouraged to discuss these findings with someone from the other side. So like I worked with MDs and we talked about some of my Zika research, and then I got to sit in on their neuro ID rounds. That was super helpful for me. And then I think, DVMs and MDs are generally the two most kind of represented fields in this idea of kind of One Health translational biomedical research. But when we get into some of the larger picture larger picture issues, of which there are many, I think we can work together to enhance our collaboration with those outside the medical fields. So one area I think this kind of One Health paradigm is lacking is anthropologists, particularly for working, in a global health context, ecologists. So Lily and I are, I think, the only MD and DVM person, MD student, DVM on the Verena team. And it's a lot of ecologists, but also, computational folks and zoologists and economists, even when you're looking at how you're going to implement some of these solutions. I mean, it's important to have those folks on board too. So really, I think if we can help provide DVM and MD students a better way or a more comprehensive understanding of public health and translational biomedicine, while also working with other fields and even, stretching ourselves, breaking down even more silos to help kind of contextualize some of these research questions and results in a larger context. I'd like to, my acknowledgement slide is usually like, size 10 font because I have so many people to thank, but thought I'd keep it short and sweet today. So the Verena Consortium, Colin and Greg and everybody in that group has been tremendously helpful. Lily, thank you, Lily, for inviting me and to your group for hosting, and then everyone at Colorado State University. And then, sorry, that's a bit grainy there, but hopefully you can read my email and Twitter and my websites. Feel free to get in touch if you are interested in this stuff or you want to brainstorm how you might be able to get involved in some of these kind of, translational or interdisciplinary efforts. I love brainstorming with folks and talking about this stuff. So please reach out. I would love to talk to you, and thank you again.
Thank you so much, Anna. That was fantastic. I think out of respect for your time and everyone's time, we'll just ask you maybe one or two questions. So John, are you still here? Do you want to ask about CP?
Yeah, can you hear me?
Yeah.
Hi, thank you so much for that super interesting talk. I guess the question was originally about sort of the similarities between tuberculosis presentation in animals versus humans, but I guess maybe more broadly, how similar in general is disease presentation, but maybe particularly infectious disease presentation in animals versus humans? And is there more, I would assume there'd be more similarity between closely related species versus not closely, but that's purely an assumption based on not all that much knowledge. So I'd love to hear more about that.
Yeah, that's a great question. And we could talk about that for a long time because there's, I hate the answer, it depends, but it depends. I think you hit the nail on the head. Generally, I mean, for species that are closely related, we see more similar disease presentations. But I mean, there are certain pathogens like tuberculosis that is fairly promiscuous. It's not terribly host specific, and it causes very similar lesions in all of the species that I've seen it in. So, the kind of caseous lymphadenoma, caseous granuloma, things like that. Viruses tend to be a bit more host specific. And so, in terms of asymptomatic presentation, we might see a bit more of that with viral species. But it really depends. And, but it's a good question. And I think, I don't want to make any generalizations, but I feel like for like bacterial infections, maybe we see a bit more parallels in clinical presentation than say for like viral species. And again, that gets to that idea of, asymptomatic versus just an animal that's actually impacted. So yeah, sorry, I probably didn't answer your question, but it's a good question. It's a hard question.
Well, thank you so much. And it definitely gave me things to think about. So appreciate it.
Thanks, Anna. So the only other question I got as a DM was, who do we reach out to if we're super interested in this work? And I would say, based on Anna's slide, that you could definitely email her. It's anna.fagre@gmail.com. Feel free to reach out to me. I'll put my email in the chat. I think Colin is also in the Zoom, which is really sweet of him to join, but
Colin's amazing.
Yeah, Colin's amazing. Colin's the head of the Verena Institute, one of the co-PIs. So definitely, I can also drop the link to our website in the chat. Oh, Colin put a little smiley. But yeah, we're all super interested in these topics and in discussing more. So I'm always open to emails, and I think Anna and Colin probably are as well.
Yeah, y'all are lucky to have this group. I mean, folks like Lily and who else was helping lead this group?
Kala, she had to.
Kala. Yeah, I mean, groups like this, I helped lead our One Health group in vet school, and we tried to bring MDs in, and I wish we would have been able to bring more MDs in, but honestly, student groups are so good for networking. And that's how you end up landing summer internships or getting research gigs or getting put in touch with somebody who totally blows your mind and changes the way that you think about everything, right? So I don't know. Yeah, definitely reach out, and I'd love to chat and reach out to Lily, reach out to Kala, and we can all work together to help you find something that might, pique your interest, depending on what your more specific interests are. Cool. Kendra, over at EcoHealth Alliance, they do some really cool work in this area too. Thanks for joining, Kendra. Yeah, I guess so that's, just reach out. Don't be afraid to reach out. We're happy to help. And yeah, I guess I can put my
Thank you so much,
email in there too.
Yeah, we can just drop all our emails. And yeah, if anyone is interested in being on our listserv, I will happily add you. Just so you get all the messages about upcoming talks. But this was really great. Thank you so much for joining, Anna.
Awesome. Thank you so much for reading all the comments. Thank you so much, everybody. Have a good day.